Well, thanks everyone for coming. Can you guys hear me all right? Is my mic levels okay?
I can hear you fine.
Perfect. Okay, so this thesis defense is on the titles Drosophila as a model for Allodynia and Hyperalgesia. And we might as well just get right into it. So I just want to get on the table kind of a definition of pain that we can work with to kind of understand what we're all talking about. So this is a nice little demonstration of how, so you have these peripheral inputs coming in of some kind of noxious stimuli, and you're going to have this obviously going up into the brainstem, lower brain, and then eventually, the cortical regions. And the IASP, the International Association for the Study of Pain, has gotten a pretty robust definition on the table for us to kind of help us guide our discussion of pain. So it's an unpleasant sensory and emotional experience associated with or resembling that associated with actual or potential tissue damage. So that's actual or potential is kind of something that's surprising to learn about pain, is that it's not always something that you usually associate with a painful event. And also, and if anyone wants to chime in or anything, just feel free. Don't be too shy. Another definition within the chronic pain that we need to talk about is two things that I'm studying. It's allodynia and hyperalgesia. So allodynia is going to be you respond to events that were previously unpainful as painful now that you are injured. And then hyperalgesia is you're responding to painful events that are normally painful, but you're responding at a much higher rate, or you elicit a much more exaggerated response than you would normally. So to kind of get an example and some examples out. So if you're injured, light touch on your arm, so you have a broken arm, light touch on your arm, that's not normally painful, but when you've broken your arm, that light touch is going to be painful. And that's an example of allodynia. And an example of hyperalgesia is if I were to, pinch the area where you say have a burn, that pinch is normally painful and you'll still usually, jerk away from it. But in this case, you're going to be making a much bigger reaction to that because you're already injured. So as far as cost, chronic pain is actually surprisingly, it's really dispersed across the population. So it's the third most important, important cause of disability and disability adjusted life years. So the cost in terms of dollars is about, $500 billion. It affects about 100 million Americans, about a third of the population or so. And that's not all that the ill effects of chronic pain have. Obviously, there's a growing opioid crisis going on, and it has at least something to do with that. So about 2 million Americans have opioid use disorder, and about 10% of people who misuse prescription opioids become addicted to opioids. And if you look at the incidence rates within the general population of addiction, it's 6%. But in the cohort of people who experience, chronic pain, that's a bit higher, and it ranges anywhere, depending on the study, from 2% increase to as much as a double increase. So it seems to have some kind of role in forming this addiction, and really getting people exposed to opioids initially. But it's not exactly clear. So it depends on the animal model. If you look at sciatic nerve ligation models, it actually makes reward less in rodents, whereas arthritis pain increases consumption for rodents of opioids. So it's not exactly clear the exact relationship between injury and reward, but there seems to be some kind of, tenuous evidence that it has something to do with it. So a couple things that we know about chronic pain, especially in humans. So you can have two kinds of these. So chronic pain is just long-term potentiation. It usually happens when most people refer to it, they mean central nervous system, especially in humans, they mean central nervous system, long-term potentiation. But it can happen in the peripheral nervous system. Example of central nervous system modulation that's going to be important for us is going to be GABAergic disinhibition. And this is caused by neurons that are inhibitory that link up into the central nervous system dying. So if you have inhibitory inputs, neurons dying, you're going to get a heightened response just because you don't have that inhibitory input. On the periphery, you can just simply get increased translation and transcription of the ion channels. And that's an easy way to increase the signal where it starts before it gets to the central nervous system. So either way is a form of long-term potentiation. Some of the stuff, so on the genetic side, we have some genome-wide association studies coming out that have linked various genes up to particular phenotypes of chronic pain. Ultimately, the GWAS data for neuropathic pain is not very good, but some pretty large variances have emerged. 18% of variance in risk of type 2 diabetes is an example of how GWAS can really give us a lot of explanatory value. Real quick, 3 problems just that are preventing GWAS from really making really good data for neuropathic pain. Obviously, sample size, all GWAS need to have huge sample sizes, and those are hard to come by. The definition, so there's many ways you can get neuropathic pain, many onsets that can cause the same pain state, but it's not really clear how we should combine those cohorts together because we can get them in many different ways. Is the same end result, is that one, should we categorize those all in one cohort, or should we split them up? And if we split them up, we're going to have a lot more cases, right? And then the last one's the definitional one. There's just not a set definition of what, how to define neuropathic pain. The fourth one's, it's not really specific to chronic pain, so it's not super important. It's more of a problem with GWAS in general. So here's kind of the, these are really, it's not super important that you focus on these, but these are the pathways that can kind of started to emerge that have been prospective genes of interest for neuropathic pain. They go across broad kinds of systems, even, metabolism and immune response, you don't really think is being contributory to chronic pain, but these studies are just trying to get at the, the obvious genetic relationship between, most phenotypes and the genes, because we know from twin studies about the heritability of neuropathic pain, even something that's, fairly dependent upon the environment, like injury. In the case of chronic pain, there's large heritability, and about 37% is what we've seen from the twin studies. So trying to find these pathways and prospective genes is kind of an emerging trend with GWAS. So clinical solutions are problematic because, people can become addicted to opioids. Not only that, if you look at the top 10 most lucrative drugs in the US, they only have a clinical efficacy of 1/4. So the reason for this is kind of related to what I was talking about with GWAS. You're going to have a lot of genes that are different, right? So you're going to need to start having personalized medicine for chronic pain and neuropathic pain. That's no different. And kind of a way that my kind of project is kind of trying to kind of help this problem mostly has to do with the third one here. But the first two are, so you're going to get bad reporting data from subjective reporting of pain and the collection of pain. And then you're going to have noise from the placebo effect. But lastly, to test these genes, you're going to need really good animal models to predict these phenotypes correctly and experiment on certain drugs. And all of that's going to be, inherently noisy. So if you don't have a good animal model that can have, sufficient numbers in them, you're going to have poor clinical outcomes because you can't really robustly test the genes or drugs that you're trying to test. So some of the big things that are unknown is, we have patients that, there's variance in who develops neuropathic pain from the same injury. 30 to 40, 50% of diabetic polyneuropathy patients develop neuropathic pain. So not everyone is going to develop neuropathic pain, even given the same insults. But generally speaking, larger nerves tend to induce chronic pain a bit more, and we're not entirely sure why that is, but I don't want to spend too much time on this for sake of time, but just so everyone can kind of have a basis to understand the experiment. So opioids, they're opioid receptors are GPCRs. So they bind agonists to the receptor sites. So the ultimate effect is a cascading molecular event. These aren't ionotropic receptors. They don't open out to the cell, although they can have that effect downstream. So they're going to get bound, and then they're going to have a cascading event, which ultimately is going to lead to inhibition, downstream inhibition of neurons. So this is essentially what we're talking about with opioids and their effects. So you're probably wondering, when do the fruit flies come into this? So early analysis on invertebrates from this study have they analyzed some tissue studies. Didn't really find anything that looks obviously similar to opioid receptors. And then even later studies have shown that there's not really any obvious candidate for receptor homology for opioids, but it's kind of at odds with what the early literature has found in the animal models. So this is kind of the first stuff you can find on invertebrates and morphine. So this is a mantis shrimp. These guys are infamous for kind of punching out of their aquariums. So the idea of this experiment is to electrocute these guys. They have a probe in their mouth, and what's going to happen is you're going to test them without opioids, and then you're going to inject them with morphine, and then see what happens to the response. And you're trying to see how long it takes to get to a fast tail flick, which is this 0.22 seconds. It's this dashed line. And these graphs are not very great, but the great takeaway from this is that it turns out that even for these knee-jerk reaction in these animals, morphine seems to increase the number of shocks and the amount of electricity needed to generate a tail flick of this speed when compared to control animals. So moving on, same lab, an experiment where they used a praying mantis and kind of the same contraption. Here they're using a lot less electricity, and they're trying to get this praying mantis to elicit a defensive reaction. And the way they do that is they give it very small electric shocks. And the same thing, they're going to compare this reaction to a reaction when they are administered morphine. What they saw was that you have large inhibition of this defensive reaction whenever you give them morphine. It takes a lot more voltage to an integrated curve too. So you can see the two bars here kind of where they're maximum 1 hour after they injected them. These are the large doses, full inhibition of reaction. And then here you have, about 1/4 of a reaction of the defensive behavior. Moving along, so a bit more relevant, a bit more modern. This is in 2019. So the idea here is that if you take a fruit fly, adult fruit fly, and you amputate its leg and you put it in a hot plate, you can compare its response to control flies that are uninjured, okay? So it's 38 degrees Celsius. This is not normally that aversive for control flies. So what they're testing here is Allodynia. Remember, this is not normally painful. So if you get a pain response in injured flies, you've induced Allodynia because the control flies are not really reacting. So what they found is it takes a couple days to onset, but you do get flies. The independent variable here is jumps. So you have the hot plate, you have the injured flies. the injured fly is going to be jumping a lot more. If you look at this graph, it'll jump anywhere from 3 times a minute, when controls jump less than, about half a jump a minute. This is pretty robust. It continues for 21 days or more, and they were wondering, what causes this. This is going to be important for later. They can show the speed at 38 degrees is going to be the same for injured and uninjured flies. Just keep that in mind. They're wondering what's causing this. So they had their theory was that it's going to be this GABAergic GABAergic neurons going into what's the equivalent of a central nervous system flies. And that's exactly what they kind of show here. They have staining right here where this is 0 days after injury, and they stained these neurons here. So 2 days after injury, you can see how the cells are dying. Eventually, we'll have no staining and no cell. So to show that this isn't, they hooked up the contralateral arm to a stimulator, and they showed that limb is also will react quicker than control flies that are not injured. So it's not that the limb is injured, it's that the inputs going into the central nervous system, those require less stimulation because the central nervous system is excited because there's less of these inputs going in. And basically they showed that if you prevent this cell death, you don't really get this heightened sensitization and then the contralateral limb. And these are just the response latencies for the limbs. And this is the prevent GABAergic cell death. You're not going to have these increased response latencies that you see. You're not going to have allodynia. Yep, if you prevent it, you don't really have any significant differences here. If you don't, you do. So why fruit flies? They're cheap, genetically tractable. They have highly conserved pain pathway, nociceptive pathways, the TRP channels, and they're very high throughput. You can have hundreds of animals when it would take the same amount of time. In rodents, you would not get, even 100. So electroshock, I'm using electroshock in this paradigm, so we can, easily in the arena split the noxious stimulus right down the middle, and you can have a choice where the animal can obviously escape to the side that's not shock. So to track the fly, we're using a camera, and it's basically just X and Y coordinates. We're just doing captures, 1 a second, and you can run multiple flies in the same paradigm, and then you can get anything you really want. You can get, speed, anything you can really calculate with X and Y coordinates. And this is just kind of to get you guys an idea of what the paradigm looks like. I'm going to fast forward here. So basically, the left side is the shock side and the right side is the non-shock side. It's kind of divided somewhere in here. In the fly, there's going to be a microscope lens over top of the fly, and it can't really climb on top of it. And you can see the escape behavior kind of starting. When it goes over here, we'll get the shock and then it'll start to escape. And we'll get the shock and then it'll start to escape. So that can kind of give you an idea of the setup. And this is kind of how over time, you're going to see the trend and the side preference. So this is the side that's going to get the shocks. This is the side that is non-shock. And the higher these peaks is the more flies that are over here. So this is just to kind of give you a general idea of how quickly the flies make it their way over there. So basically the setup is that they're in the arena. Every 2 minutes, the shocks are going to increase by 5 volts. The experiment is going to go until 100 volts, and the experiment runs for 44 minutes. And we have the position of the fly the entire time. We know how much volts every 2-minute interval is being administered. So we have a very clear picture of what's going on the entire time. So want to get to the results. That's going to be in the way. Okay, so there's a couple assumptions that we're working with here that I want to get on the table, so I don't feel like I'm, p-hacking here. But so for Allodynia, we have to control the volts because we have to look at specific voltages where the stimulus just is sub painful. So to do this, we look at controls, we see what they're not reacting, and we make the principal justification that if they're not reacting at this volt and they are reacting at the next step of voltages and controls, that means we should look at the previous voltage in the amputated flies. So that's kind of how we find what is not painful to the fruit flies. And then we test those voltages for Allodynia. For males, the controls react at 15 volts and the amputated react at 10 volts. So that's a way that we can look at the data and say that that's where we don't really know where this is going to onset, but it seems to be setting on here. So female controls reacted about 15 or 20, so their threshold is a bit higher, oddly enough. So I looked at look for Allodynia in both 10 and 15 volts. For hyperalgesia, pretty much everything beyond anything that's painful is on the table. So 10 or 15 volts, but there's some initial preferences for the arenas. So to kind of account for that, we included 0 to 50 volts. And the reason we chopped off the 55 to 100 volts is that so it just starts to cripple the flies and they can't really escape too well. And we don't really see these modulations. They don't increase the overall response of the flies. They're going to just increase the onset of the pain. So it's not increasing the total threshold that they're going to respond to this stimuli. So there is a top end to these modulations, and we're not really interested in those. So we'll talk about that more a bit later, but oh, I forgot I added that slide. But okay, so these are the analysis from the ANOVA. So we did one-ways and we did a four-way. So preliminarily, it looks like, so we put into a data frame, males and control males and males that are amputated, and we looked at, so we're controlling for the sex, we're controlling for the voltage, and basically the time, because we're controlling for the voltage. And we found some significance for male amputated flies when we compared them to control flies. There seemed to be a side preference that sets on earlier than in the control flies. And we didn't really see anything for the females with the one-way analysis, but there is an effect that's linked to sex we found on the four-way analysis. So to kind of picture how this looks, so this is how the data is going to be looking. So on the x-axis here, we have volts. On the y-axis, we have the probability of shock. So the higher the bar, the more they prefer the shock side. The lower the bar, the less they prefer the shock side. So as it goes down, you can see how it trends down with the increase of voltage. Sounds like it's exactly what you should expect. The more painful the stimuli, the less you should see them entertaining that stimuli. So for the males, we're gonna look at the males first. So you can see here how the males, like I said, they don't respond at 10 volts, but they do start to respond at 15 volts. And over here, the third bar, as you can see, now these are confidence intervals, they're not standard errors. You can see how the it bends downward, and to get a closer look. So I'm going to overlay these two to kind of give you an idea. And once we do, we can see that there's a gap here between the confidence intervals. This isn't super informative, but you can see how the gray bar is the controls and the red bar below it is the amputated ones, and there's a gap there. And indeed, with the ANOVA, we see that there's a significant difference at 10 volts here. Same thing for the females. So the controls over here, we have the amputated here. When we overlay them, we can see that at every voltage past the painful voltage here, there's going to be a difference between the controls and the amputated. The amputated are going to prefer the non-shock side much more than the controls. And we didn't get a single voltage where there was significant difference at one voltage, but when we looked at the whole voltage from 0 to 50, we can see that there's collectively there's an effect. I mean, there's only one spot where the means are in the opposite direction. Okay, so I think I'm pretty much right where I want to be with time. So effects of morphine. So these are one-way ANOVAs here, and I'm going to do the same thing. So it doesn't appear that there's any direct effect of morphine without an amputation. What I mean by that is if we put morphine in the food of the flies, we conduct the experiment in the same way, but the only effect we have is that these seem to be actually shorter. The bars seem to be shorter, meaning they're preferring the non-shock side a bit more after they're administered morphine. And we don't really see any effect in females, really. Not really anything to speak of on the ANOVA side. Not really any effects on probabilities. There's some effects on activity that we'll talk about in a minute, but there doesn't seem to be any simple direct effects on the probability of shock in either sexes. However, when we look at the mixed effects, so these are all amputated animals, both male and female, there does seem to be effect. So both of these right here are the ones that we just saw. So these are the same animals that you guys just saw. And then these are the new animals that are same procedure done, same amputation, but the only difference is they have access to morphine in their food. And we do pick up, so females, they didn't show allodynia, but they did get fairly close to being statistically significant differences with the one-way ANOVAs at the 10 volts and 15 volts, but it's not significant. So it does seem to have some, it shifts the means, but it's not enough. But for the males, it does appear that it actually reverses the allodynia that we saw at 10 volts. And I'll show you again, I'll remind you. So here's 10 volts. We have these are confidence intervals, I'll remind you again. So this is statistically significant. And the blue bar above here is the males on morphine. And we can see that it doesn't quite look like a control, but it's higher than in the amputated. And it is statistically significant at this voltage. So looking at the females, so this is what the hyperalgesia looks like. These voltages are painful to them, and the controls show a higher preference for them. If we look at amputated ones, this is the same overlay. The blue bars above the red are going to be where the means of the females on morphine and amputated are. So while there's no direct effects of morphine, it seems that the means are significantly shifted when they're injured and administered morphine. It seems like they will entertain more of a shock than their only amputated counterparts. So this is the results of the four-way ANOVA. I don't want to talk too much about this because there's a lot going on, and I'd rather take some questions, but really quick. So there's not that many direct effects besides voltage, and obviously voltage is going to have statistical significance. But amputation kind of the effects of amputation depend on sex, and that reach significance. What that seems to kind of illustrate is that Amputated males, they do demonstrate allodynia, but they don't demonstrate hyperalgesia. And it's actually the reverse for females. Females will demonstrate hyperalgesia, but not allodynia. So the females are the ones that are having an effect here. The amputated females are the ones that are driving the significance levels, just because for every voltage that they have 15 to 50, and those are all significant, whereas the males just have the one voltage level that's significant. Effects of sex also depend on morphine, which is kind of surprising. It seems that the males kind of get, their preference goes way down. So when you give them morphine and their control fly is not amputated, their preferences shrink in comparison to controls. So they seem to be hyperalgesic with morphine, whereas females don't really seem to show much of a difference either way. So this kind of raises the question, like, what is this actually doing in the fruit flies? So there's been some people thinking about this for quite a while. There was an experiment done where they administered morphine to crabs, and it didn't seem to actually make them more willing to make trade-offs where they wouldn't, crabs like dark areas usually. So if you put a crab in the middle of an arena that's light, and they'll usually try to flee to dark areas. If you administer a crab morphine and you put them in the arena and you're trying to get them, and you administer shocks whenever they go into the dark arena, you would think that if they were on morphine, they wouldn't really care if they got shocked, and they would just go to the preferred area, the dark area. But it doesn't really seem to be the case. They don't actually, when they're on morphine, they don't actually willingly enjoy the shocks just so they can get to their preferred environment. But Elwood kind of thought about what this means. He kind of concluded that morphine might be inhibitory on pretty much everything, including nociception. So not just nociception, it makes them unable to respond. It doesn't just reduce nociception, it's inhibitory to most of the systems in the organism. And it's kind of hard to rule this out because we do see activity data that is pretty consistent with that hypothesis in males. So I'm going to kind of explain what's going on here really quick. So this is the same x-axis we had where there's voltage over time, and the y-axis here is activity. So these are pixels traveled per second. So it's not a real distance, but it is a very good stand-in. So this is how much the flies are moving. So activity between the males amputated and males control, or let me back up. So controls are going to always exhibit the most activity. That reads true for females too that are not amputated. But also males are going to show generally more activity than females. So what this kind of means is that obviously amputation is going to reduce activity, but so does morphine. So if you look at this line and this line over here, the gray, are they going to be the controls, right? And if you compare the controls not on morphine and not amputated to the controls on morphine, what we see is there's a marked reduction in activity almost to the point of amputation. But when we see amputation and morphine, there is a reduction of activity, but it's not huge. It's not as big as it was. Furthermore, the activity doesn't have really anything to do with the one-way ANOVAs, and the reason we know that is because if we look at, for the males, if we look at their activity here and we look at their activity here, the groups that are doing this calculation are the amputated not on morphine and the amputated on morphine. So these comparisons are almost exactly the same activity levels. They're a little bit different in the confidence level, but they're exactly at 15 pixels per second. So the reversal doesn't seem to depend on activity. So obviously activity has something to do with the amount they're moving around and probably the amount they're going into the shock side. But for this calculation, it doesn't seem to depend on it really much at all. So in females. So this is why it's kind of hard to kind of figure out what's going on, because we don't see this effect of morphine in females. So when we ran an ANOVA analysis on activity, we really don't see that there's much of an effect on activity for females. The females that are given morphine don't really demonstrate that much of a difference in their activity. It's pretty much the same graph. And same with the amputated. There's not that much of a difference either way. Here's our ANOVA table for activity. Obviously, direct effects of amputation and morphine on activity, but there's also a mixed effect of sex and morphine. So, like I said, males have drastically decreased activity as a result of morphine administration. And again, the effects of amputation and morphine on activity. Amputation status depends on morphine status. So kind of the main questions I want to kind of wrap up here so I can take a lot of questions. Not really sure why there's sexual dimorphism between the males and females for allodynia and hyperalgesia. The primary literature that just came out in 2019 also didn't find any hyperalgesia in males. So I have to probably conclude that they had the same problem. And I'm not sure that they did females, but I know they did males. So they didn't find hyperalgesia in males. And if they would have tested females, maybe they wouldn't have found allodynia. But what are the exact effects of morphine? We seem to have a pretty good case that it does something, it has some modulatory role when flies are injured, but it's not so straightforward when it's a direct effect without the injury. And lastly, do injured flies consume more morphine than non-injured flies? This is also another question. So we didn't actually record the amount of food that they consumed when they have the morphine mixed in. So maybe the obvious question is, well, how do you know how much was administered? We don't. We do have another paradigm to look at that, but not for this. But we know from honeybees that generally speaking, amputation doesn't really induce increased morphine consumption. And could be a number of reasons why that is, but at least challenges the assumption that they can have some kind of an appetitive formation with morphine when they're injured, even though it might have some modulatory role.